Hello, and thank you for coming to my talk. My name is Michael Murphy, and I'm a PhD student at MIT, where I am co-advised by Professors Stephanie Jagelka of Computer Science and Ernest Frankel of Biological Engineering. Today, I'm going to present our work on using self-supervised learning to classify immunohistochemistry images by cell type specificity. In short, this work's contributions are threefold. We developed an effective representation learning algorithm for immunohistochemistry images. We combined this representation with an independent single-cell RNA dataset to learn a classifier of cell type specificity without requiring human annotation of images. And we demonstrate this approach better assigns known proteomic markers in kidney to their correct types as determined by human pathologists than approaches relying solely upon single-cell transcriptomics. We start with some high-level motivation for our work, which arises in the context of experimental design for multiplexed immunofluorescence. Multiplexed immunofluorescence refers to a suite of related technologies, all developed in the past few years, for measuring protein expression in situ at subcellular resolution. These approaches all entail staining a tissue sample or cell culture with antibodies complementary to target proteins of interest, and which have been conjugated to a fluorophore, and then inactivating the fluorophore and repeating this staining over several cycles, permitting simultaneous imaging of up to high tens of different protein targets within the same sample. Because it directly captures cells in their native spatial context and does not require tissue dissociation, multiplexed immunofluorescence can tell us a great deal of information about cellular communication and microscopic tissue organization that is lost with technologies like bulk proteomics or single-cell RNA sequencing. Multiplexed immunofluorescence also tends to produce visually striking images such as the ones shown here, which are of an experiment measuring 50 markers in human lymph node. The main difficulty in designing multiplexed immunofluorescence experiments is the selection and validation of an antibody panel. Although the literature continues to grow, to date the number of antibodies demonstrated to work with CySIF, one of the more popular implementations of this technology, numbers only in the few hundreds, and is biased towards well-studied targets such as immune cell markers. There therefore still remains a great deal of trial and error involved in developing a reliable panel, and this can take weeks to months of experimentalists' and pathologists' time. Among other modes of failure in the antibody validation process, one that should be amenable to machine learning solutions, and which we choose to study in this work, is the fact that the standard practice of nominating marker candidates on the basis of transcriptional specificity to a particular cell type, which we can measure readily by a single-cell RNA sequencing experiment in the same sample, is not guaranteed to give rise to reliable proteomic signals of cell type. This can be simply due to unavailability of a validated antibody for the chosen marker in the tissue of interest, but it can also potentially arise due to biological processes, such as post-transcriptional regulation that can decorrelate RNA and protein expression. And recent studies measuring both modalities at the single-cell level, such as the one shown here, indicate this decorrelation can even affect genes known to be canonical markers of cell identity. The Human Protein Atlas provides a resource orthogonal to single-cell RNA sequencing for design of multiplexed immunofluorescence experiments. This is a large-scale compendium of proteomics assays that includes a dataset of remarkable scale, a repository of approximately 10 million immunohistochemistry images. Each image represents a circular tissue punch that has been stained with a brown antibody, plus a blue nucleic acid counterstain, and this has been carried out for approximately 30,000 unique antibodies in biological triplicate across 44 different healthy human tissues sourced from several donors. The downside of this remarkable resource, however, is that it lacks fine labels of cell type specificity at the resolution we're interested in when doing multiplexed immunofluorescence. For example, this randomly selected image of a stained kidney sample indicates expression only in either glomeruli or tubules. In contrast, single-cell RNA experiments in kidney have identified many tens of different cell types in that organ. So we cannot use this data directly to read out a list of antibodies to measure. It still requires visual interpretation by a human expert to determine exactly where the antibody signal is localizing, and pathologist time is costly. To state the problem explicitly, the object we desire here is a mapping from an antibody, represented here as an IHC image and nominally targeting some gene, to an indication of whether it's going to be a suitable marker for some cell type of interest. If we had a subset of images labeled at our cell type resolution of interest, as opposed to just those two coarse regions from the previous slide, we could set this up as a standard supervised learning problem. However, we cannot generally assume this data is available. All we assume we have here are these unlabeled IHC images and this noisy but readily available proxy for cell type specificity at the protein level, which is specificity at the transcript level, as measured by single-cell RNA sequencing in an independent dataset. Self-supervised learning has emerged as a paradigm for learning effective classifiers using large unlabeled datasets. Compared to supervised learning, which presupposes the availability of a large dataset labeled by human experts for some target task of interest. In self-supervised learning, we design a pretext task, which is also a supervised learning problem, but one for which labels can automatically be generated from unlabeled data without human input. Examples of such pretexts might include imputing a masked out patch from the rest of the image, colorizing a color image that was artificially reduced to grayscale, or recognizing whether one image is a rotated copy of another. Solving this pretext task then yields a pre-trained encoder network. To solve the target task, we hold this encoder fixed, and we retrain only the final classifier layer on a smaller subset of the data for which we do have labels. The principle here is that if we choose a sufficiently informative pretext, this encoder should learn a representation of the inputs in which the classes in the downstream problem are going to be linearly separable. And it is much more sample efficient to simply train a linear classifier on these than it is to learn the entire model from scratch. Contrastive learning is a fairly general and particularly successful formulation of self-supervised learning. The intuition of contrastive learning is that we should seek a representation in which semantically similar positive examples in our data are placed close together, while semantically dissimilar negative examples are placed far apart. But in the self-supervised setting, we lack direct knowledge of the semantics of our data in the form of class labels. Contrastive learning instead synthesizes its positive examples through data augmentations. For example, inverting an image of a cat does not change the fact it is a cat. Negative examples are likewise generated by just sampling uniformly at random from the training data. This works if there are many classes, as there often are in image recognition tasks, as the probability of drawing a semantically equivalent example to the query becomes small. And to make some of this intuition more concrete, this is the loss function we minimize in contrastive learning. We draw a query example X from the data distribution P. We sample a single positive example from a positive distribution conditioned on that query. In the typical case, this is a data augmentation of the query image. And we then sample a batch of B examples from a negative distribution, which in fullest generality, we might also condition on the query. And then we minimize this softmax loss term, where we favor large inner products between positive pairs in the numerator and small inner products between negative pairs in the denominator. The art of contrastive learning that we exploit in this work is in the design of these positive and negative conditional distributions. Specifically, the gap that we address in this work is that ordinary image augmentations, which lead to successful self-supervision on computer vision benchmarks such as ImageNet, do not represent meaningful variability among semantically equivalent immunohistochemistry images. Effects like stain discoloration, changes in morphology due to age and health of the donor, or even damage to the sample during mounting are far more complex than simple rotations, crops, or hue shifts, and these would be quite difficult to fully characterize and implement as data augmentations. We solve this problem by exploiting the replicate structure inherent to most biological datasets, including the Human Protein Atlas. Rather than trying to synthesize biologically plausible positive examples from a given query image, we simply draw our positive examples from any image stained for the same nominal gene target. Because these are generally drawn from different batches, donors, and sometimes even stained with different antibodies entirely, we naturally encapsulate sources of variability in IHC by doing this without having to explicitly characterize or engineer them. We observe this scheme does indeed induce clustering structure in the embeddings, as shown here in the middle UMAP plot, and the mark of a successful contrastive learning procedure, whereas the naive approach using image augmentations alone on the left does not. However, we also observe, shaded by color here, imbalance in the donor labels across each of these clusters. This is undesirable, and we later found it reflects a bias in the rates of co-occurrence of pairs of donors within genes in the dataset. So we're actually just recovering a bias inherent in our training data. To fix this bias, we therefore additionally modified the conditional distributions of negatives to only draw from the same donor as the query. And this does induce uniform mixing of donors across clusters. We quantify the effect of both of these procedures via cross-validation accuracy of a logistic regression trained to predict the donor label, and find that both of our modifications confound this classifier, indicating that we've induced a desirable fairness property with respect to this attribute of donor label. Now, to understand the origin of these clusters in our embedding space, we associated each gene represented in our data, which comprises around 10,000 immunohistochemistry stains in healthy kidney, with its mean expression across 12 cell types as measured in an independent single-cell RNA dataset, and which we've indicated via shading each dot. Each dot is an IHC image, and we've shaded them according to this cell type specificity measurement from this single-cell RNA dataset. And the result of this is remarkable. It reveals we have learned an embedding in which clusters correspond to major anatomical regions and cell types in human kidney, solely through optimizing the self-supervised objective we described earlier, and without ever requiring a human expert to actually interpret staining patterns. Now, having learned an informative representation of IHC images that preserves biological semantics and is invariant to histological variability, we now return to our original problem, which is how to derive a classifier that sends a representation of an antibody to a cell type, using only the data we have available to us. And to solve this problem, we now make use of the noisy labels available to us from an independent single-cell RNA dataset. More concretely, for a given gene, we compute its mean expression for each of K cell types that were identified via clustering this RNA dataset from Muto et al., published in Nature Communications 2021. And we then normalize this mean expression in each of the identified cell types to unit length, so you get a vector in the K simplex. And then we train a multi-class logistic regression model to predict this vector in the K simplex of cell type probabilities from our learned embedding of an IHC image stained for the respective gene. So even though this transcriptional label is only going to be a noisy proxy for the proteomic signal that we actually want to predict, our choice of restricting the model family to a simple function of a proteomic representation biases the classifier to predict what we actually desire, which is proteomic specificity, rather than overfitting to transcriptional noise. To evaluate our performance on the marker selection problem, we found a paper in which expert pathologists manually annotated IHC images of 130 cell type markers from 4 types in kidney. We then set up an evaluation in which we compare our model's predictions of cell type specificity to those derived from various tests of differential expression from single-cell RNA sequencing data, which represents current standard practice for nominating marker genes in a multiplex immunofluorescence experiment. We find overall our predictions, which are derived ultimately from integrating unlabeled immunohistochemistry with single-cell transcriptomics, better agree with expert labeling of immunohistochemistry than relying solely upon transcriptomics. To summarize, in this work, we developed an effective representation learning algorithm for immunohistochemistry that translates a state-of-the-art computer vision approach for self-supervision to real biological data. We then combined this representation with noisy labels derived from an independent single-cell RNA dataset to yield an image classifier without ever needing human experts to annotate images. And as a specific case study, we showed this approach better recapitulates cell type specificity annotations by human experts in kidney than approaches that rely solely upon transcriptomics. We finally note this work has some limitations in its current state. In this publication, we only investigated one organ, namely kidney, and we are presently working on scaling the approach to the entire human protein atlas, which comprises 10 terabytes of images across 44 tissues. We are also working on moving the approach from the proof of concept implementation demonstrated here to a more broadly usable tool, which will be a plugin for the open source multiplexed immunofluorescence analysis pipeline, MC-Micro. That concludes my talk, and I thank the audience for their attention.